right, everybody, in the interest of time, we're going to go ahead and get started. Let's welcome Ben. Take it away.
Thank you. Hello, everyone. I'm Ben. I'm coming from the University of Otago. I'm a combination of different departments along anatomy, biochemistry, and immunology and microbiology. I have everything but less of anatomy, so it's a really weird combination of department. I'm going to talk today about biosecurity deployments of CRISPR-based environmental biosurveillance assisted by artificial intelligence. So the most interesting thing that I've been working the last 3 to 4 years, counting my PhD, if I go for my undergraduates, I have been working on it for the last 7 years. We're working on biological invasions, invasive species, threatened species, and mostly biosecurity concerns or threats. So there is a very interesting number on 2023 where we see that there is a joint report of 49 countries that they report that the only biological invasions they account for 423 billions of economic losses. And that's massive, taking into consideration that we don't even use a 10% of that losses to try to manage that massive input on the production systems. So another thing that just came out this year is the World Economic Forum. They are at the long term, 10 years on the future, we're going to have a lot of issues with extreme weather events that I think that we are already seeing a lot everywhere. You have seen Chile and the wildfires over there. You should check it out. They are massive. The second one is the biodiversity loss, which is really linked to biological invasion, critical change to Earth system, which is like all interconnected, and they are a biosecurity concern for New Zealand and also internationally for many, many, many countries. So the too long didn't read is basically that the invasive species generate this massive pressure on natural and productive ecosystems. And it's really important to understand that if a natural system is affected and lost all the services that they provide, most likely the productive ecosystem that they depend on, the natural ecosystem, they are going to fall behind too. So it's kind of like all this interconnection that we need to keep safe and secure from biological invasions and biosecurity in general is the natural but also the productive ecosystems. So here where it enters the CRISPR-Cas toolbox, where many people knows about the gene editing power of this technology, but also CRISPR can work as a diagnostic tool. And it's pretty easy and actually works in 2 or 3 steps. So you will have a Cas nuclease that you are looking right here on the A, on the top corner. And then this caster team, when find the target of the Cas nucleus, when find the target, is going to cut a reporter, and you will have a fluorescent signal. Easy, just like a qPCR. But then you can use the same probe of reporter, and then you can make like a lateral flow readout, or better known as a pregnancy test, where when you have 2 lines, you have a positive. When you have 1 line, you have a negative. So it's pretty straightforward, very easy, isothermal, and you can get results in like 20 minutes when a qPCR can take like 2 or 3 hours. So in 2019, I thought that this was a really great idea, which failed. It wasn't a complete failure, but it kind of took like 1 year of development. It was a really time-intensive and repetitive, a lot of sequence information to analyze, which was really difficult, especially because I was just an undergrad with a little of luck, and I got some kind of funding that I could play with, and I failed to actually get all this working properly. So I was like, okay, what now? So I was talking about this technology with some people in New Zealand, and they were like, why you don't do a PhD here about this technology and you try to develop it better with what you already learned about this failure, this experience, right? So that's where the artificial intelligence comes in and start integrating, because I was very lucky that in 2022, when I started my PhD, the MIT released a public knowledge artificial intelligence that they trained with 19,000 guide RNAs. So now all the things that took me 1 year, now an artificial intelligence could do it in 5 minutes, which was a massive increase of power on research. So a little bit of the overall things that this artificial intelligence does is that it can align the sequences. It will identify some kind of consensus on all the mapped single nuclear polymorphins, and it will start finding these high coverage regions, and it will try to design guide RNAs that they also have a predicted activity. So all of this happens really fast. So for example, if you're working with a single gene, it will take like 1 minute to run it, and you can run it in this same laptop. Then if you want to go for mitochondrial genomes, you can also run it in a laptop. And then if you go for viral genomes or bacterial genomes, you can also run it in a laptop. But then if you go to bigger organisms like microorganisms like a dolphin, for example, you will take a server to run it. But anyways, the most important thing is that you can streamline with artificial intelligence and also make a better decision making in the process, because it ranks the prediction of these guide RNAs and how they are going to most likely work in a diagnostic environment. So we started to work on this. We created a system which was based also from the MIT, from Gutenberg 2022 and 2019. But basically, it's a 3-step detection system that all happens simultaneously, but it's really quickly, and it's quite interesting. So you have a DNA amplification, which happens with the RPA, which is recombinant polymerase amplification, which is an isothermal, just like the PCR, and it runs at 37 degrees Celsius. So at the same time that you are amplifying the DNA, you will have a T7 transcription generating RNA template, and at the same time you are increasing the RNA template, Castor's team will start detecting that sections. And you will have a fluorescent signal, or you can also have a lateral flow, as I said before. So that's what we call the Sentinel platform, all the mixture of this artificial intelligence, then the CRISPR-Cas technology, and then how we deploy it in environmental DNA. So this is all came together and came as the Sentinel, which is the platform that now we are developing with many, many partners that will show afterwards a few results that we got till here. So the Sentinel platform has an artificial intelligence base. So it's scalable and versatile. So we went from marine invasive species, then we changed to rat species, and now we're detecting dolphins, avian influenza, cowrie dieback, and many other different targets. So this kind of got really, really broad. So now we are calling it like a platform of detection for environmental DNA. So one of the other quality of this platform is that it's really specific. It's also highly sensitive, so you can get down to 10 attomolar or 5 copies per microliter really, really quickly with not much of an effort with artificial intelligence. So you have quick results. We usually call it an hour, but it does is actually how much we run the reaction. But usually in the 10 attomolar, we are seeing reactions that they become positive around 20 to half an hour. So we actually don't need to run the full reaction, but this is just to see the kinetics and have more data to keep training the artificial intelligence. And the cost effectivity of deploying these assays is $3.5 USD per assay. That's still fairly high in comparison with a qPCR, but you have to consider that you can field deploy this technology, which qPCR cannot. So that's where it starts to shine. And also, which I will show you too, is that the CRISPR-Cas technology has a high resistance to inhibition, so you don't need to purify your samples that much, which in qPCR you need to purify your samples lots to make it work and make it work optimal. So a little bit of the proof of concept. The first one, like I said, which was marine invasive species. There are 2 model species in New Zealand. They are both invasive. They arrived really long ago. So it was easy to actually obtain samples and environmental samples. So we have Undaria, which is wakame. So if you go to Otago Harbor, you go to the sea and you see this brown kelp, that's Undaria, shouldn't be there. And Sabellus palazzani, which is in the North Island. So we took a few environmental samples, and as you can see, I think this is the pointer. Is it? Yeah. So as you can see, we have the positives where they should be positives, and we have the negatives where we have the negatives. And we got some like pristine samples that they should be negative for both, and they stay negative. So that actually showcased the proof of concept that this technology can work on environmental samples with high specificity. So then we were like, okay, but Undari and Savela, they have this massive difference between these two species, so it wasn't like a really challenge to get something specific working on. So then we moved to rat species, where we work with the Department of Defense of the United States. Don't ask me why they want to detect these rats. But then the interesting thing about these rats is that they are really similar in the mitochondrial genome, which is the most abundant when you want to get a sample of them. So they have like 3 to 4 SNPs of difference. We were actually able to detect the 3 species really specifically, as you can see on the picture. I didn't show all the data, but basically we have activity here, and then we have another set of guide RNA that they work for Norvegicus and Ratus ratus here. And we actually went a little bit further, and we're actually showing that it can work in 2 different populations of the same species across US and New Zealand. So then we were like, okay, what else? What is the next challenge for this stuff? So we also got an MBIE smart idea with the same methods of Sentinel. The interesting section of this smart idea is that we have the Macrocterium bovis. It's a species complex of bacteria that they have 2 SNPs of difference. So we are now going from 4 or 5 SNPs of difference with the rats to only 2 of them. And we actually also, with the same technology and same platform, we're able to make some kind of detection, differential identification, even if you see that there is a little bit here of activity that's really, really low because we're working with millions of copies of DNA, which is usually not the case in clinical or relevant samples on the environmental section. So it was like, okay, cool. So there's actually this work in environmental samples. So we contacted some people from South Africa that they work with bovine tuberculosis. They have lots of it. And the interesting section about all the samples that you can see here is that they were Sanger sequencing, they throw gene express ultra, they did also qPCR allele specifics to certain genes of the bacteria, and we had 100% of which one they were supposed to be positive and the ones that were supposed to be negative. So that's pretty nice because we were expecting, especially with the Mycobacteria species, because there's like 100 species, we were not sure if we will get this really highly specific detection as we wanted, but we could. So that showcased a little bit further what this technology can do with the CRISPR-based environmental biosurveillance. So then we were like, okay, what's the next limit that we can push? So there is some discussion about we don't want to keep extracting samples in the field detection assays because that takes a lot of time, but also years that you have to carry with you. So then we were like, okay, so people want to work with this kind of raw samples. So can we actually throw the CRISPR-Cas system just to detect in raw samples with what the artificial intelligence told that they could potentially do? So we just went to the supermarket, purchased a little bit of milk, spiked with genomic DNA of bovine tuberculosis, and we see that we can actually get fairly good, we can get fairly good detection down to -4, which is around 42 bacteria per reaction, which is very meaningful, especially for environmental samples and especially for New Zealand standards where bovine tuberculosis it should not be there. So you should always have a negative. So that's why we go so low. So we are completely sure that we have a really low amount of bacteria around. So then a little bit of the take-home message about all what I have been saying across the CRISPR-Cas technologies and everything else is that the artificial intelligence can accelerate and streamline the biosecurity deployments, but not only that, I see it like it can save cost and time, especially when you are trying to deploy an assay and you can take like 1 year, like I did 9 years ago, and now with this Sentinel platform and the artificial intelligence, we can do it in 2 months if we are lazy, and we can do it in 2 weeks if we have the money and the time to actually run everything as we would like to. So a little bit of this crazy idea of the biosecurity and the potential of CRISPR as a next generation detection platform. We sent this to Transient Biotechnology, which just got published like 2 months ago because it's a really trendy idea that they like to publish. So I would like to acknowledge all the partners because I haven't done this work alone. It has actually been a lot of collaboration with the CIP, US Department of Defense, USDA, the Cochrane Institute, of course the University of Otago and the Ministry, the MBIE that we all know here. And also I have an amazing people working with me and amazing scientists of all these people and way more of other people that we work with every day to make this platform better and trying to push to a real platform that New Zealand and internationally we could use to face the biosecurity threats that we know, but also the emerging ones in the next coming years. Thank you.
Do we have any questions for Ben?
Yes.
Thanks, Ben. Very cool. Two questions. One is, if you tested multiplexing with different dye colours, would that work in principle if you had multiple targets in the same?
Yes, it works in principle.
And the other one is just about your hit rate. So it looks great. How many times did it not work that you used an AI designed target?
So that's a very good question. So I have deployed around 12 different species, and it will depend on what extent you want to make this detection work as you want. For example, for the bovine tuberculosis, we had to generate 10 diagnostics assays, and 2 of them hit as we wanted. So we basically failed 80%, and we got a 20%. But for the rat species, we got it on the first go. So I think that's kind of, it depends how big is the challenge and how many SNPs you have for making this differential identification, but it's way faster what a human could do on the same time. No worries.
Thank you very much. Incredible. Can you use that software for designing, obviously for the guides for the CRISPR case, but can you just use it for designing normal primers for amplification, like consensus primers?
Yes, you can. So you can do qPCR with them. So it's benchmarked and done with qPCR alongside, but also you can do RPA with them. So you can do qPCR and RPA and the guide primer if you want to.
So you could use them just for designing normal PCR consensus primers?
Yes.
Thank you. Let's give another hand to the speaker. Thank you so much. Great job.
Thank you.
Great job. Okay, that top one, yeah. All right, our next speaker is Kiel Hards. Let's give it up.
Yeah, good afternoon, everyone. My name's Kiel Hards. I'm a senior data scientist at a company called EPI. I'll tell you a bit about them in a second. A bit of background from me for those that don't know. I sort of came from Otago originally, did a PhD in microbiology, and then from there moved into the Ministry of Primary Industries, where I worked in the biosecurity space for a while, and now I work here at EPI. Before we get into that, just a few acknowledgements for the talk in general. So the project I'm going to tell you about was funded by the Ministry for the Environment, and as part of a larger program of work that they're doing. Also thanks to a number of other organisations, WilderLab, eDNA sequencing laboratory, amongst other things, the Global Biodiversity Information Facility, you'll learn a lot more about them, and Department of Conservation, Hawke's Bay Regional Council, and now Earth Sciences New Zealand, so that logo's a bit out of date, who has been involved in providing data that we'll be talking about. So who are we at EPI? So EPI is a multidisciplinary team combining a number of people from different domains. That's biology, data science, software engineering, and essentially the whole idea of the company is we try to connect data, science, and people for a variety of projects. And that can range amongst a number of things, a number of different clients, sectors, as well as a number of different things that we do, whether it be in infrastructure, software development, dashboards, you name it. If it's in that space, we have a lot of interest in talking to people and seeing what kind of challenges we can help them with. So that's who we are. Let's talk a bit about environmental DNA, which is why everyone's gathered here today for this particular talk. So environmental DNA, otherwise known as eDNA. So what is this? Some of you may be familiar, some of you not. You know, the entire idea about eDNA is this is something that we can use for detecting organisms in the environment, right? So there's usually some kind of genetic material that's been shed from various kinds of organisms, you know, primarily those that are familiar, maybe more in a higher organism space, but it can be all kinds of sources of DNA. And what it is a really powerful, non-invasive way to monitor, you know, presence of organisms and relate that to other kinds of parameters. Really particularly powerful in its biodiversity and biosecurity space, right? And so something that, you know, particularly the Ministry of Environment wanted to improve was our sharing when it comes to this biodiversity data, because we collect a lot of it in New Zealand, but the large-scale data sharing is difficult. And I'll talk a bit about why that is. Some of these are technical barriers, and we'll get into that a bit. And it really limits our ability to reuse, share, and make this data visible for the greater benefit. And so this is something that the Ministry of Environment wants to improve as part of a larger program of work. And what we'll talk about is one particular aspect of that project that we were funded for by MFE. And really, the overall question behind this, you know, the thing that we wanted to improve is how do we improve and address these technical barriers by balancing large-scale submission of these complex genomic samples whilst also maintaining those checks and balances for data integrity and quality validation, right? And so, you know, this is what we ultimately produced in this project, and this is what I hope to introduce to you today and tell you a bit more about, is this tool, this open source R package we developed called eDNA Bridge. Essentially, it's this R package that you're able to use, and the overall aim, is in the title, is to act as this bridge to, you know, help a variety of, you know, people, users, organizations, to take what they may have from these eDNA samples, which, as you will see here, typically sequenced by some kind of sequencing laboratory, probably performing usually a method called metabarcoding, to identify all the organisms in a sample that you may have taken that can, with these eDNA traces that you're identifying. And then we need to get that into certain data repositories that are publicly available, and there's a couple of them. You may be familiar with this one, GBIF. You may also be familiar with other websites like iNaturalist, these websites where you can go on, you can see this biodiversity, typically occurrence data, where someone can maybe have gone out, taken a photo of a bug, here's the bug, and you have a verification, it's this kind of species, all of that, but instead of, say, photographs, we're doing this from the eDNA data, so occurrence-based eDNA data. And what we're aiming to address with this package was bulk or automated data uploads to really help, you know, eliminate those barriers that are stopping people from getting to A to B, because it's just maybe too complex, too repetitive, too time-consuming, and really just, you know, ideally the end goal is you can just have a one-click button, click, there you go, your samples are now public, and it's now everyone's able to use it, reducing that need for, you know, they're reducing that manual burden when it comes to this process. And so as we'll go through, this package actually supports many ways of using it for many different kinds of people. You get very guided workflows, get very, you know, very interactive, help work you through this process, which can be quite technical for your first time, and walk you through it. Or for those that are more familiar with coding and want to, you know, have a more customized workflow, you can do a lot more of that, a lot of functions that you can bring in and build them into these automated pipelines. And one thing that we're particularly aiming at was for sequencing laboratories that are performing this service. So in this case, we worked a lot with Wilderlab. So this is a, you know, Wilderlab provides this metabarcoding as a service. They're an accredited laboratory, and they've done the majority of the work in this space in New Zealand. So let's talk a bit about how you actually submit a sample and where we kind of aim to address the bottlenecks. So this kind of schematic here that you will see, we kind of have a number of steps, right? You take an eDNA sample from the environment, maybe it's a waterway, you collect water and you'll process it using some kind of sequencing kit, probably involves syringes and filters. You know, I'm sure many of us are familiar with that kind of work. And then it needs to be identified, right? This is where the sequencing laboratory comes in. They'll do the sequencing, identify all the different taxa in a sample, maybe relate that to some kind of counts or some kind of metrics. A particular thing that WilderLab does is they take a lot of these observations and they relate that to their internal models to tell you things about water quality, health, and stuff like that. But from there, then we need to get those tables of information, which can be quite complex, and then we need to then do some standardization and formatting, and then we need to actually upload it somewhere. And this is where it gets a bit complex, right? Because then we need to do things like standardize all the DNA samples that we have. Then we need to build archives in particularly rigorous formats that are aimed to be, you know, very rigorous and help standardize everything that we're doing, but takes a bit of expertise to become familiar with, right? And then we need to actually upload it to particular institutions. And the way that GBIF works is you upload these data into various different servers that can house this data. Typically, think of it like a franchise, right? You might have a lot of different McDonald's that are around that can be owned by different people. In this case, GBIF, you can have lots of different servers that hosts the data. And then the data is made available centrally to GBIF through a crawler that accesses and indexes all the information on said servers. And the one that we worked with was actually through a server that was hosted and maintained by the GBIF NZ regional node, but people can set up their own ones. And then finally from there, as I mentioned just before, that's indexed by GBIF, and then you can search it on the GBIF website, search it up there and see all the information. And so where we really aim to try and address these technical challenges was here in this step metadata provider and data uploader, right? Because this is something that is very technical, and the collaborators and stakeholders involved identified as a real pain point in that process. And this is the part that we wanted to ultimately automate, or at least improve the efficiency of with this package. And so that's ultimately what we did and what we set out to do. So what I'll do at the end of this talk is show you where you can get it. But here's a few details about how we use it. And I can tell you a bit about a pilot project that we undertook. So using eDNA Bridge, so one of the things we wanted to highlight is that we've built a lot to help people guided through it with documentation, command line interfaces, all the standard things you might expect with an R package. So if you're using this for the first time, you're like, I really want to upload samples to GBIF, but I haven't done it before. You can work through, some of the documentation we have. You can spin up a documentation website using standard commands that you would use for an R package from the package down library. You can also see here we've got an eDNA Bridge command line interface, which you can use mainly intended for one-off submissions, and obviously that's not something that you use in bulk, but as you get familiar with the process, you might transition to a more automated approach or workflow. And then what we've done is we designed it in a way to be very modular, so that if you wanted to extend it and add new capabilities, it's very easy to do so. And so we have these modules of data ingestion and validation, the metadata and creation of these archives that we call Darwin Core archives. That's a specific format that your data is prepared in for submission to GBIF, as well as the upload of said Darwin Core archive, and ultimately the actual publication, automating that publication process in the server itself. So you can actually, take with credentials, obviously, you can take credentials for the sequencing laboratory, download from WilderLab, have it automatically prepared on your own computer, and then with credentials for whatever server you upload it for, that process is also automated. So essentially a one-script process to then have your samples appear on GBIF. So in order to both demonstrate the functionality of this package, but also to kind of launch the MFE program to start getting more of this biodiversity data actually uploaded and publicly available, we undertook a pilot project which involved a number of participating organisations and in essentially organising data between them and walking them through not so much the technical process, but that organisational process of approving and understanding what we want to release. So the organisations that participated in the pilot was Department of Conservation, Hawke's Bay Regional Council, and Earth Sciences New Zealand. So what we did is we took all of their samples that were listed as public in Water Lab, with a few exceptions. Hawke's Bay provided a numerical list closely matching what is listed as public, just so that we had that double checking in there. So essentially, all public samples that they had through that database in Water Lab, and we took it through a custom workflow scripting approach, which we then take some examples of how that looks and we put in our documentation. And essentially what we're able to do is, you can see here, upload over 4,500 metabarcoding samples from Water Lab, which represents about 765,000 occurrence data. Just think of that as rows in a table with also extra information as well. And ultimately we were able to do that in a matter of minutes. So normally this kind of process for, single uploads could take maybe an experienced person half a day. And if you are not experienced, well, it wasn't happening, right? That's why we took this project in the first place. So what we're able to do is to almost design this fully automated approach. Obviously in this case, if you find errors in the validation module, you obviously go back and fix them. But in the best case scenario, you can have this process fully automated, and that's what is available in the package. So with all that developed, obviously there's a few things to think about moving forward. So one of the things that when we developed this package, we actually kept it very general so that we can actually use it for a lot of different types of data. It doesn't have to just be eDNA data, it could be general occurrence data that's formatted in similar ways. It's all, at the end of the day, we're uploading multiple types of occurrence data. If it's that kind of data format that works with it, which has led to endless discussions about whether we should call it eDNA bridge, GBIF bridge, something else bridge. I think we've all agreed that bridge is part of the package, but I think ultimately we will likely settle on the name eDNA bridge. But what that also leads us to is a bit of an idea of, we can actually take the package in more directions in the future. And so some of the things that, could be possible is extending, say, the ingestion module of our package to incorporate new types of data standards for eDNA. One particular one is called the FAIR standard, FAIR-E, so findable, accessible, interoperable, and reusable. So it's more like an upgraded version of what I mentioned before, this Darwin Core standard. Other things that we could do is support other types of servers that can host information. So GBIF have made a newer type of server called an MDT server, Metabarcoding Data Toolkit. I mean, if it makes sense of the name, that's more focused on eDNA. Also, Ocean Biodiversity Information System, where we could all support all kinds of things and tease it in that direction through that modular nature. But also a few considerations as well. Obviously, we can automate the technical process, automate the upload, but what we can't do is automate the organizational approval process. So if you want to upload samples to GBIF, your organization still needs to be registered with GBIF. You still need to go through that process with GBIF to register that. You still need to find a place that you can host your data. You still need access to that. And obviously, if you have metadata that you don't provide to sequencing laboratories, such as overall project staff and personnel, you're going to need to figure out a way to provide that. And we provide mechanisms in a package to easily ingest that, but you still need to know where that data lives to be able to feed it into the package. And obviously, all sorts of governance decisions are outside the scope. And, whilst we can validate data is, valid or not, we can't validate it's factual. That'll be on the people using it to ensure that that is the case. So just as we start to get to wrapping up, so the package is available on GitHub, but actually we've been able to host it with GBIF themselves. So GBIF International, GBIF Secretariat have put this on their GitHub page, and EPI will be the maintainers for now, and then we'll move forward with that into the future. So GBIF/eDNA bridge. This is actually technically the first time we've kind of talked about the package, so we quietly uploaded it, and on a webinar that we're doing this Friday with Water Lab, we'll talk about it more, and we'll talk about another medium soon. But for those that are interested, feel free to take a look. It's open source now. Cool. So hopefully that's told you a little bit about how some of the work that we do can connect eDNA with global insights, public accessibility. So yeah, just to wrap up. So yeah, we're EPI. We love dashboards and open source data science. We don't talk about dashboards today, but we do love it. And we love to talk about amazing people we love to talk with amazing people and the data that they get to work with in the process. And just a last little plug, we're also looking to expand and grow our team. So if you know anyone that might be interested in roles or careers, particularly in intermediate or junior developers, please talk to me, my colleagues Petra and Nick are here tomorrow, or you can also reach out directly if you're interested. Thank you for listening. It's been great having me. And yeah, feel free to come and talk to me over the course of the day if you have any questions.
We have time for a quick question or two. Does it also help uploading the sequence data?
It can do uploading the sequence data, although the data set that we used didn't contain the DNA sequence, but we support the DNA extension that would subsequently allow it.
Nice.
To GBIF, not things like NCBI.
Really nice talk, thank you. It's just we can chat later, but I wonder, are you aware of the local context GBIF integration work around?
Yes, this is a great question. So yeah, you know, the Local Context Initiative is GBIF are developing an extension. It's not fully released yet. So in some cases with the upload, we actually added extra data around iwi area of interest in other extensions. But when that's formally released, we're interested in seeing what that kind of changes to support that would be. So yeah, now we've talked a lot with different parties about that, and we hope, you know, once that's fully released, it'll be interesting to see how the package can be extended, yeah.
Thank you very much. PDF? Yeah.
Okay, so just summarize that. Just go back to it.
That's it, but in the browser.
So we just need to go to full screen.
Ah,
And
thank you. All right, our next speaker is Ben Marmont from DairyNZ.
Cool, thanks everyone for coming out. As mentioned, I work for DairyNZ, but I'm also a PhD student at Queensland University, and my co-authors obviously helped along the way. This presentation is based off of a conference paper that was given last year at the Australasian Dairy Science Symposium, and you can find that paper by searching the title of this presentation or on my ResearchGate. So a bit about DairyNZ. We are a industry good organisation, so we do a lot of research, extension, advocacy for the dairy sector. We're funded by a levy, and you might also notice the little watermark in the corner. So a bit of a disclaimer, AI help with these slides. So this presentation is around the journey of that DairyNZ has been on and is continuing through in modernising the way we do science. And so that is done via the Modern Science Workflows Project, which is, I guess, our internal name for the project that's funded and this work sits under. So I guess the motivation behind starting out on this journey is that the quality of data that we're getting when we do research, and also outside of the research area, but that farmers are getting as well, is just growing exponentially. And that's the case in many fields. But, like you or I might wear, a Garmin watch to tell us how we're sleeping and moving and breathing. We can also get things like that for cows. So collars, but also ankle monitors. You can all sorts, things in the shed, things in the machines that they eat from, all generating, data every second. Or if you're taking methane measurements, that can be even more frequent. What's happening in the feed that the cows are eating, how are they processing it, and then outside of the farm, data that's being generated from satellites as well. So what does the grass look like, how is it growing, how does water run across the land? So massive amounts of data and, kind of running into boundaries in the way that our scientists would traditionally deal with it. And so that is kind of the wall in Excel, right? We're a pretty science-based organisation without, historically huge capability in big data and infrastructure and people. And so, we started this process to go, how do we deal with 20 gigs of data when we're used to dealing with 20 megabytes? And so the journey we've kind of been on is pretty well reflected in this diagram here, which we've lifted from that paper. And so, when we started out, we were quite impaired. There was perhaps a few people doing data science, but, very, very specific and isolated and perhaps not the most well documented or certainly not best practice. And then, as we've developed along, there's become, I suppose, specialised hubs or teams, like data science and modelling, for example, but still quite isolated and not that well, interruptible with the other parts of the organisation. And then we kind of realised, well, hey, we're not really at where we should be, so how do we get there? Started having aspirations about how we can get better and how we can improve. And that, includes leadership coming on a journey as well. And then we start getting towards the stage we're at now, and perhaps if you ask my co-authors, they might take other views on where we're at, but, we're getting to that stage 4 or that stage 5 where we've got some capability and we've got development plans and we kind of know where we want to go, but there's perhaps still some work to do. And so if you're around tomorrow, my co-author Mubashir will be talking about spinning up some tools pretty quickly in quite a trusted environment. And so that might give some insights there if you're interested. And so thinking about how this project will take us to where we want to go. So, with the goal of wanting to modernise the way we do science and handle data, like internally the way we went about getting there is kind of around these 3 themes, I guess. Perhaps the deliveries of the project if you want. So there's the infrastructure piece, how do we get the tools that people need to go on that journey? And so for us at DairyNZ, that's Snowflake as the cloud storage solution, and then for tools, it's POSIT. So you'll hear in a second, a lot of it has been about R and getting, a fundamental capability across the organisation in R. But that's also with the POSIT tools. So POSIT, on your laptop, POSIT Workbench if you need a bit more grunt, but then also connect to deploy whatever you've produced. So then the capability piece, so running courses called R for Data Science based on the textbook of the same name, and then internally as well, giving people, I guess, some motivation and a cohort to go through the journey with. So we have courses that we sponsor people to go on, but also community meetings, people who are working on R, we can go and we can thrash out our problems or we can share our wins, just build a bit of a cohort for this journey. And then the third piece is business discipline. So are people documenting the code the right way, storing it in the right place with access to the right people, etc. And so what this kind of is a visualization of what I mentioned there, but that central part of the diagram comes out of the R for Data Science textbook that I just mentioned, and then the coloured pieces around it are the way we do each of those steps. So the data comes in from Snowflake. You might tidy, transform it, visualize it, model it. You might iterate that a few times. That code to do that would be stored in GitHub. You might do that on your laptop, but if you're running out of grunt, you might scale it up and do it on the workbench. And then once you've created your report or your app, then it goes up to Connect, and then that can be set to be public facing, or it can be, behind credentials if it's for internal use only. So that's the kind of workflow that we're working with now, and it seems to be working pretty well. So some things along the way. So that R course that I mentioned, we started off teaching it internally, and that kind of worked, but when it's just internal, you can kind of lack a bit of buy-in, and people might struggle to actually claw out the time they need to do a course, because it is essentially, a full semester-long course. So we've farmed that out to the University of Waikato. Auckland Uni, we are a Waikato-based company, so that kind of makes sense. But this year will be the third year of that course, it is open for enrolment now if anyone did want a kind of one-off course for that. So third iteration now. And we've helped design the course. I taught the second iteration of it, so we've been quite involved in making sure that we're getting what we need out of the course and the uni, and so it's been quite a cool collaboration and a reasonable commitment for us. So for each of these iterations, we've sent 10 people or more on that course. And then building on that course, we have run our stats courses internally. Those ones are perhaps not as well attended, but for the people that need it, we can build on that capability. I guess one of the nice things about going to the uni as well is people kind of know, hey, it's a couple grand and there will be a grade associated with it, and so you're kind of a bit more motivated. I'm an economist, and we know people respond to incentives. No one wants to fail a course. And then coming back to that R community meeting that I mentioned before, so that's a place for both internal R users and external, and heck, even data science people to come along, share their wins and learnings. And it's not quite been monthly. While I was running it, it kind of ended up more bi-monthly, but it all comes out in the wash. So some real-world, examples. I mentioned some of that wearable sensor data. So there's been a few papers in the last couple of years looking at the heat stress cows are experiencing. So when it's super hot and they're getting all that sun on them, actually cows' production is inhibited, and so what is the sort of link and what actions might you take to try and mitigate that? And so there's a couple of papers where they did just that, taking data where there's super granular readings across multiple animals and multiple locations, and then taking it from this huge piece of data which you wouldn't run on your laptop and turning it into something usable. For the economics team, we've, I suppose, been champions for the use of R and some of these tools, and there are two publications that we put out each year, one Dairy Statistics and one the Economic Survey, and so they're kind of, they come out every year, and those are now completely done and are hosted in POSIT, and they are viewed thousands of times a year, and so that's been a really cool journey, taking that from a document that was the analysis was done in Excel and then written in Word, and then it goes to the marketing team to make it pretty, whereas now that happens completely in R. The data's ingested, the publication's made and booked down, and then published in Connect, and so that's a real win and something that we're quite proud of. This is, the extension on it, the open science we're kind of moving towards, but I might gloss over this one today. And then, so moving towards some of that more best practice stuff is, we're still quite actually early on in this piece. We've still got a ways to go, and that's why I kind of think we're not quite at that fifth stage I mentioned before, but how do we make sure that if I get hit by a bus coming home from the conference today, that my colleagues can go and pick up the chapters I write in Dairy Statistics and publish it like I was never around, right? So how do we really make it resilient? And so we're in the process of setting up our GitHub organisation. I know many of you probably already have that, but for us that's quite new and a journey we're working through. We've got our single sign-on set up, and you know, with Snowflake and with the GitHub, so that's working quite nicely. And then Kyle mentioned the FAIR principle, so that's something we want both within, you know, the data coming from Snowflake, but also the repos, so that, you know, we're not having people creating code that already exists in a separate repo, and that it works together, etc. So I suppose as we're kind of winding down, one of the things that we're kind of reflecting on is just how important data science is becoming, and perhaps, you know, to the traditional science organizations, that perhaps there shouldn't be a team that's siloed and just does, you know, the scientists run the trials and the data scientist deals with the data and then the statistician does the stats, but perhaps that those scientists need perhaps a wider skill set that encompasses data science and should be part of their training. And so just like digital calculators replace the slide rule or the abacus, perhaps scientists could not need data scientists anymore, because, you know, everyone has some of those skills. And obviously there'll be exceptions for the super tricky stuff, but perhaps that's something, the vision we could move towards. Oh, I thought I had one more. So DairyNZ is, as I mentioned, it's funded by the levy, so we would like to thank the dairy farmers of New Zealand for funding, I suppose, DairyNZ, but also this bit of research which allows us to improve the way we work and take us on a journey. And so there are a few people that were central to this project, which is now, I guess, in its 4th or 5th year, and so their names were up there. But, you know, there were people from LIC, there were people from Waikato, from Lincoln, and BSI, you know, that have just come on this journey with them. So thank you to them. That's me.
Do we have any questions?
So I see you collecting all this data, and as a genomics person, I'm like, are you bringing that over into breeding values, or any future plans?
Breeding values are set by a subsidiary of DairyNZ, New Zealand Animal Evaluation Limited, so
Okay.
I probably wouldn't want to talk to those.
All right.
So I'll just kick that to touch.
I was just thinking when you said the production goes down when the heat is up, I'm like, oh, that would be a great signal for choosing, you know, the next few generations.
Yeah, and I know there is research into that. You know, there are some slick genes running around cows that deal with heat a bit better.
Yeah.
So that's something you could look into if that's what you're perhaps interested in and didn't know about.
Yeah, I'm interested. I just need data.
So there's lots of geospatial stuff in this we don't feel background too deeply into Slack. ArcGIS is getting built certainly, and QGIS being largely Python based. Are you touching into any work in that space, or is most of the stack in R and
Yeah, so the starting point for data science at DairyNZ is definitely R, but there's room within there for other tools. And, you know, there are people through the business that use, you know, Python, that use ArcGIS. And so it kind of depends on what the specific use case is. But, yeah, there are, you know, people that are doing a lot of their work in ArcGIS, for example. But as I mentioned, we're still not all the way at that journey. And so how do we, you know, one of the things we still need to do is bringing in those people that are using different tools and languages and helping them come on the journey with us and getting everything in the same place. And I think we have some analysis coming up that's going to be some more geospatial stuff. And so that will be the question for us. You know, do we try and do it in R, or do we actually pivot? Do we go to something Python-based, or do we go to ArcGIS and park the POSIT tools for now?
Kiel, I think, actually does some stuff in geospatially. What's the package name? You know? Is it Leaf or is it?
We can talk about it after.
Yeah, okay, sorry.
Like, we have done some, we do some spatial stuff in AHA already, but not anything overly complex, I'd say. Quite rudimentary. Yeah.
From the work that you're doing, are you generating digital products as such? So you're taking data and you're turning it into something that eRangahau might be able to interact with on a day-to-day basis. So are you productising the work you're doing?
Yeah, so if I think about my example in the economics team, there's three major things we put out. There's those reports, so one comes out in June, one comes out in December, and so that they come as data from DairyBase, our database of farm financials for the season, and data from processors and a few other things to create those reports. They get made into book down books, web pages, or Shiny apps, whatever, and then they get linked to on the DairyNZ website. So yes, they are, and they're publicly available. The third one is the DairyNZ Econ Tracker, and that one is an interactive dashboard that's a Shiny app, and it's hosted on Posit Connect. And so all of these are, they're not open source, but they are publicly available. And so the open source thing is something we've been thinking about, but the data would never be able to be publicly available, just given the agreements. And so, perhaps we're not sure if there is how much value there would be in making the code open source if the data is never available to run the code. But it's something we've thought about and is perhaps a nice idea for the future.
So you have quite clear controls in your environment about who owns the data.
Yeah, so people can put in requests to get dairy-based data, but there is a process there, and there's got to be pretty good reason for unaggregated data to go externally, because it is financial data of 2,000 dairy farms every year for 15 years. So I guess, yeah.
All right, let's thank Ben again. Thank all our speakers, and let's go get some tea.